Hello everyone. Welcome to the next microbiology course. This is a review for people who are going to take the technologist and technician exam. I will point out some key things that you need to know based on if you're taking a technologist versus a technician exam. In general, most of all the information covered, technologists need to know. There are a few things that technicians don't necessarily need to know. With all of the classes, you'll see general slides show up, and those general slides are going to be telling you what to expect, what the objectives are. Everyone should have gone over the first PowerPoint in this course, which goes through every last objective. Those objectives are taken directly from the exams. Therefore, these are items that you need to concentrate on. There's a lot of information in microbiology. When I teach microbiology at the colleges, we teach it over a course of 24 months. And it is 24 months of 5-hour classes with 20 hours of homework. So it's a lot of information. I'm going to try to narrow it down. There will be some information that's background information, just so that you can have as a base for knowledge, for understanding, not necessarily something that you're going to be tested on. This first session is about defining terminology associated with bacteriology. I'm not going to go through all of the terms as you have this information, so you should be able to print those out. I recommend making flashcards, and as you make flashcards, you can write on the front aerobic and flip the answer on the back. You can download apps to make flashcards. There's lots of tools out there to where you could quiz yourself. There's Quizlet. So there's different ways for you to get the information and kind of keep going it over the information constantly. I will stop here and say this about cytoplasma and cytosol. Those are two terms that sometimes get confused, but they are not exactly the same. So cytoplasma is the chemicals and structures within a cell, and cytosol is actually fluid, pretty much compounds and ions within a cell. So they're in there, but it's more the fluid. Sometimes people interchange those two, so just be aware of that. Facultative aerobe and facultative anaerobe. Just remember facultative meaning easy, something that is able to do, and then you have to know aerobe is for oxygen, anaerobe without oxygen. Flagella. Pay attention to the fact that flagellum, the LUM, is a singular version. Most of the time in microbiology you see flagella because bacteria normally have more than one flagellum. However, there are some that only have one flagellum. When it gets to the temperature, I'm going to go over some of those in another PowerPoint and a little bit more detail. Temperatures are very important because that is used to identify organisms. You may have a case study and it says this organism was found growing at 37 degrees, or this organism was not able to grow in 42 degrees. So that becomes extremely important as far as identifying bacteria. I'm going to stop on phagocytosis, process of engulfing and destroying microorganisms by neutrophils, monocytes, and macrophages. I'm only stopping on this because I know that there's some of you who may be taking hematology at the same time as you're taking microbiology, or you may be going into hematology next. And just remember, this phagocytosis process is going to become critical in the hematology world when we talk about antigen and antibodies and what happens in the body to cause the antibody formation. Okay, here's the basis of what you need to know as far as identifying a bacteria. The media requirements. One of the key ways to identify bacteria back in the day, the pretty much first and only way you had to know if something was a bacteria started with media. Of course, there's a lot of things that are taking place in the world to where you can identify things, but we need media to start the process to get the bacteria to grow for most things. So the first thing you need to know is temperature requirements. Psychrophiles, they're cold loving, means they like 15 degrees Celsius. Mesophiles, some people say mesophiles, they are pathogenic organisms, and thermophiles are heat loving, 50 to 60 degrees. pH. pH has to be optimized. That means that we're going to add something into the media in order for the pH to be the best that's needed for the particular organism that we're looking for. Bacteria require a pH normally of 6.5 to 7.5. Fungi require a pH between 5.0 and 6.0. That should tell you right there that those two different type of microorganisms do not grow on the same type of media. They may start out growing on the same type of media. Okay, bacteria growth requirements continuing. Oxygen. That's in the first part of definitions. You got aerobes, facultative anaerobes, obligate anaerobes. Again, obligate is harmed by oxygen. That means that when oxygen is present, they cannot grow. Obligate means they have a duty. They must be in that state, which means in this case, they must be in the presence of lack of oxygen. And then some have the formation of superoxide radicals. They're toxic. They can be neutralized by catalase, peroxidase. And if you see a term that has ASE at the end of it, that is an enzyme that breaks down something. Okay, so remember ASE is an enzyme. Superoxide dimutase, which is possessed by aerobes and facultative aerobes. Other atmospheric requirements: microaerophiles and canophiles, which canophiles require higher levels of carbon dioxide. That's what the CO2 is there. Aerotolerant do not require oxygen, but they are not poisoned by oxygen. So that means if there's a little oxygen there, that organism may actually survive. Might not be as much of it growing on your plate as it would be if it was in an anaerobic state, but you might still find that organism. Okay, in the microbiology laboratory, when we have media set up, media has to follow certain standards. The media, first and foremost, has to meet the growth requirements, and there's general growth requirements. So what we do is use what's called agar. Agar is a sugar, a polysaccharide derived from marine algae or seaweed algae. And bacteria love to grow on it. And I'm going to tell you this because I've said this to people in the past and they kind of didn't quite understand. Bacteria, like most other things, they do like sugar. Sugar is good for them. So of course, sugar is something that helps them grow. And therefore, you see they grow on algae that has a sugar in a polysaccharide. The media melts at 100 degrees Celsius and then it becomes solid at 45 degrees Celsius. So it's a process. Then we can have different types of media. Complex media is the most common, which means it has peptones and astracs that allow it to attract different types of bacteria. So it might have some enhancements there. We have media that's selective and differential, and we'll get into that. And some media promotes growth and other media inhibits growth. Dealing with anaerobic media, remember anaerobic is lack of oxygen. So this media has reducing agents which bind dissolved oxygen, so it eliminates the oxygen. You have to remember thioglycolate. Thioglycolate is normally called thio in the microbiology laboratory, and the broth tubes must be heated up prior to use in order to get rid of the oxygen. The other thing that you have are gas pack envelopes, and the gas pack envelopes contain different things, one being Na2CO3, which is sodium carbonate, and then you have sodium borohydride. If you add water to either one of those, then it produces carbonic acid, which is the H2CO2. The H2CO2 increases the growth of the bacteria that likes to grow without oxygen. They normally have what's called a palladium pellet inside of the pouch to cause a catalyst. That's what calusize is, the reaction, which means it is the catalyst. It's the thing that promotes the growth. It causes the change to occur. And then some require hemin, which is a blood derivative. You will get into that later, vitamin K or yeast extract. Again, the requirement for the media is going to be very important because that's going to give you your hint as to what organism is growing. And all of the exams have case studies, no matter if it's technologist or technician. And each one of the exams is going to give you information such as organism grew on this particular media, and you're going to be required to know what's in that media, what type of organisms actually grow on that media. Typical incubation, 5 to 10% CO2 incubator or a candle jar. You could do a Google search and look up a candle jar so you can see how that looks. 35 to 37 degrees Celsius and 50 to 70% humidity. So they need a little bit of moisture there. The media can be selective and or differential. We'll get into that more. Another thing, basic thing that you need to know is about sterilization and inhibition techniques. Heat denatures protein, which means it breaks down a protein. Moisture in an autoclave is steam under pressure. So if you combine moisture and heat, 15 pounds of pressure per square inch at 121 degrees Celsius for 15 minutes will kill most bacteria. This is important. It's on like every test out there. What's the best temperature? What are the requirements for autoclaving? And it's always 15 pounds of pressure per square inch at 121 degrees Celsius for 15 minutes. The exam will try to trick you up and it will say something like 30 minutes or 5 pounds of pressure or 150 degrees Celsius. So if you don't remember anything else, you need to remember this because I have seen it on so many exams and students have come back and said, that question was on my exam. So it's out there. Just for a side note, QC for the autoclave is Bacillus stereothermophilus. Okay, if you break that down, stereothermo, just the thermal part is heat. Philus, Philus, is loving. So it is a bacteria that loves the heat. And that organism will grow in that environment. But if that organism gets killed, then you're like, okay, cool, we've got rid of everything because we've reached the right temperature to kill everything else out. All right, so then you have the second thing that you can do to sterilize or inhibit is a dry heat, a flame, or an incinerator. This is hot air and it's normally at 170 degrees Celsius for 2 hours. This is not normally a question other than to know the difference between what a regular autoclave is versus a dry heat. Pasteurization, which is very high temperature, this is what they actually do to the food to sterilize and inhibit bacteria. It is not necessarily sterilization, it's inhibiting, which means it is reducing how much of it, keeping it from growing. It doesn't kill what's there, which is what sterilization is. So pasteurization occurs at 140 degrees Celsius for 3 seconds. Out of all this information on this page, the biggest thing for you to take away is this first section here, and to know that pasteurization does not involve sterilization, and that dry heat is actually like an incinerator. It is not going into an autoclave. Okay? Next, how do you sterilize or inhibit something? Filtration. You put things through a filter, right? So the pore size is 0.2 micrometers to 0.45 micrometers, and that means that it's very tiny. And it's used for like sugar, urea, media, and vaccines, which, okay, right now we're dealing with COVID, And we're dealing with vaccines out there, and sometimes this is one process by getting rid of bacteria that is naturally a part of our life and part of environments, and taking those bacteria out of these things such as vaccines, so that we would not be injecting those into human subjects. Refrigeration. This is a big one because most bacteria do not like to be in the cold. Therefore, they grow slowly. So if you place something in the refrigerator, most of the time you will reduce what is grown, and some things you will completely inhibit what's grown. Desiccation means you add a desiccant that kind of dries out the water, and then that doesn't allow the bacteria to continue to growing, but whatever's there stays there. And that process is called lyophilization. So the organisms are viable, meaning that we can actually take them out of that environment, plate them onto another plate, and grow them or test them or whatever we need to do. Osmotic pressure, hypertonic solution, hypertonic means increased salt solution. It causes plasmolysis, which means killing, breaking down the plasma. This is used in cured meat and fruit preserves. So cured meat, you can think of potted meat, is kind of gross, and fruit preserves. So when you get through a micro, you might completely change your diet, at least for a little bit. And then radiation forms hydroxyl radicals, and it damages the DNA. And the last section that you need to know something about is sterilization and inhibition techniques, and it is called disinfection. Removing the infection, and that means the bacteria is already present and we're going to get rid of it. You don't have to know these in great details as far as what they do, but you do need to know examples of disinfection techniques being phenol, halogens, and alcohols. You can easily remember part of this because right now everyone is using alcohol-based sanitizer to remove bacteria and viruses from their hands. Okay, now this is a little bit more for microbiology, just for us, if you want to know, just to be kind of like extra smart, you can say, hey, that alcohol is denaturing, which means it's taking off the protein and it's dissolving lipids, the fat. So that's how it's killing the bacteria. And that's kind of just a cooler thing to know, be able to say to somebody. Okay, so you're going to go on to your next PowerPoint because your exam would not come until you finish the other PowerPoints that we have. Alrighty, see you then.